So I would like to share something about the SARS-CoV-2 pandemic that we are in. And we have been, coronavirus have been among the globe for several years now. It has first been discovered in 1931. And until 1966, this was only in an animal form, and only in 1966 the human variant was discovered, which leads to like cough-like, cold-like symptoms up till 2002. And then SARS emerged, which was a much more virulent form of coronavirus, causing acute respiratory disease, and had a fatality rate of about 10%. And then about 10 years later, in 2012, MERS emerged, which was even more virulent and had a mortality rate of about 35%. And of 2019, we're in the SARS-CoV-2 pandemic, so 7 years later. And the thing is that the infection of these coronaviruses, we do not have a real treatment for them. It can only result by means of infections control and not really by medical intervention. And most of the research topics focuses on viral replication, while there is less known about the host consequences. So to say something about that and to have an idea about how the virus actually enters the cell so you can have a better way of treating individuals, you have to know how the virus enters actually the cell and how it infects the host. And in 2005, there was a paper which showed that the SARS, so the CoV-1 virus, uses ACE2 to enter the host and also causes lower ACE2 levels. And ACE2 is a receptor, and as you can see here, the ACE2 knockout mice, I don't know if my cursor is shown, but hopefully it does. The ACE2 knockout mice, which has no ACE2, has also a lower virus titer, and also the spike RNA of this virus was much lower in the knockout variant than in the wild type variant. So this shows that the SARS-CoV-1 virus needs ACE2 to infect the host. And this Western blot shows that that the ACE2 levels in the wild type variant, when it is infected, that ACE2 levels are going down, so that you have lower ACE2 levels, where this is not the case for the ACE enzyme itself. So the infection also results in a downregulation of ACE2. So what about this ACE2? I mean, why again does ACE2 matter? And therefore we need to go to the angiotensin receptor, the angiotensin system, the renin-angiotensin system, in which angiotensinogen is converted into angiotensin 1 by renin, and this is further converted to angiotensin 2 by the ACE enzyme. And angiotensin 2 stimulates the AT1 or the AT2 receptor, and where it has a prevalence for the AT1 receptor. And this causes pro-inflammation, vasoconstriction, and increased vascular permeability, and also increased oxidative stress. Whereas the angiotensin 2 receptor causes the opposite. It is anti-inflammatory, it leads to vasodilation, and it leads to a reduced oxidative stress. And ACE2 is needed. It is a particular molecule because it is a receptor, but it's also an enzyme because it cleaves angiotensin 2 in the angiotensin 1 to 7 form, which can stimulate the mast receptor and also leads to anti-inflammation and vasodilation. So if ACE2 is downregulated or is lost, you have the path of angiotensin 2 stimulating the AT1 receptor is more pronounced, leading to this acute respiratory distress syndrome. So, and this was shown in another paper in 2005, which shows, and you have to pay special attention for the dark blue squares and the dark blue here in the graph. It shows that if you have the ACE knockout mice treated with an acid-induced lung damage, that it shows higher elastin levels lower oxygen levels and also a higher wet to dry ratio of the lung tissue. And you can also see in the picture that it showed more severe lung damage. And in the Western blot, you see that the wild type acid-induced lung damage also has a downregulation of the ACE2 again, and no changes in the ACE enzyme. But what's more important is that if you restore this ACE2, then you have less lung damage. And this was shown in this paper. You have the this will be appreciated in the green circles and bars. And here you see the ACE2 knockout mice with acid lung-induced lung damage. And if they are treated with recombinant ACE2, you see a lower amount of elastase, and you see also a lower wet to dry ratio of lung tissue. And this is also the case for the wild type animals. So also the wild type animals, which as I showed before, has a downregulation of ACE2, can be restored by recombinant ACE2. So the lower ACE2 levels, if this is the case, if these lower ACE2 levels is indeed the case, then we should see higher angiotensin II levels, because this ACE2 should break down angiotensin II. And this was shown in this animal model in which they used both the acid-induced lung damage, but also intraperitoneally injected the spike protein of the coronavirus, and they saw a downregulation of ACE2, which is shown here in the Western blot, but they also saw an upregulation of angiotensin II in this lung tissue, which is shown by the green bar. And not only for animal models, but this is one of the first papers from China showing that in humans, in patients infected with the SARS-CoV-2, so with the coronavirus that is currently going around the globe, that patients infected with SARS-CoV-2 had higher angiotensin II levels than their healthy controls. And this was also associated with the viral load and with the oxygen level, showing that the higher the viral load, the higher the angiotensin II concentration, and the higher the oxygen, the lower the oxygen levels, the higher the angiotensin II levels. And what is so important is that it's not only, this does not only hold true for coronaviruses, but also for other viruses, especially some influenza viruses. And this was shown in this paper in 2014, that mice infected with H5N1 also showed lower ACE2 levels, downregulation of ACE2 levels, whereas the ACE enzyme was not affected, and also higher angiotensin II levels. And using recombinant ACE2 restored the H5N1 induced lung injury. And this was also seen in humans, both infected with either the H5N1 or the H7N9. The ones that died because of this virus infection had higher angiotensin II levels than the one that survived. So this is actually all coming down to this summary, that you have the coronavirus, which binds to the ACE2 receptor and gets internalized into the cell, where it can infect the cell and breaks down ACE2. And then ACE2 is not there anymore to convert angiotensin II to angiotensin 1 to 7, and can lead via the AT1 receptor to the acute respiratory distress syndrome that we see. So the question is if this is all there is, or whether there is also some lack in switch from the innate to the adaptive immune response. And that's what we described in an earlier paper of ours, in which we suggest the following. When you get infected by the virus, you get an upregulation of angiotensin II, which leads to a downregulation of nitric oxide. And the angiotensin II receptor, when it connects to the AT1 receptor, stimulates many different inflammatory cells, and also leads to an upregulation of chemokines, which can cause this cytokine storm. And the switch is actually where NO either can be restored, or whether this balance between lower NO levels or high NO levels, which in the end lead either to an upregulation of the AT1 receptor and a degradation of ACE2 and further infection of the host, or whether the NO can be partially restored, leading to a switch to the AT2 receptor, more ACE2, and then more NO production and initially stimulation of the adaptive immune response. So this is probably why in some patients after 7 to 10 days you see some kind of a switch, and they go very quickly into the acute respiratory distress syndrome, while others actually recover after clearing the virus. So according to the above, it could be that angiotensin receptor blockers, the ARBs, might be a good treatment option. And this has already shown in animal models. These were treated with losartan after they had there was a lung injury by acid-induced lung damage, and they saw that the treatment of losartan diminishes the lung injury. And you can see this from the dark blue triangles and bars, that the animals that were had an acid-induced lung damage and were also injected with the spike protein and then treated with the AT1 inhibitor at lower elastase levels and a lower wet to dry ratio of lung tissue than the other animals that were not treated with the AT1 receptor. And also in an H5N1 infected model, so they infected mice with this virus, they saw that the treated mice with losartan had a better survival and also had less lung damage. And not only for humans, but there not only for animals, but also for humans, there is some data. It is a very small study and some preliminary data that treatment with telmisartan, one of the most strong ARBs, might be useful in the SARS-CoV infection. This was a randomized open label trial. They randomly assigned 80 hospitalized individuals with a SARS-CoV-2 infection, and it had to be individuals with less than 4 days of symptoms, and they randomized them to either 2 times 80 milligrams telmisartan for 14 days or standard of care. And they had a primary outcome of a decrease in CRP levels and a secondary outcome of days till discharge. And they saw for their primary endpoint that there was indeed a significant decrease in CRP levels at day 5 and day 8, and that there was also a shorter time to discharge in the individuals that were treated with telmisartan. So we recently also showed some circumstantial evidence of the relationship between ARB treatment and SARS-CoV-2 infection. And what we did, we obtained the prescription data on ARBs of 29 countries all over the world, and we calculated the first 3-week mortality slope. And this means is that as soon as there was the first report of SARS-CoV infection within a country, we calculated the amount of mortality that was happening within that country in 3 weeks, and we calculated the linear slope, and then we associated the percentages of ARB use in a certain country with the slope line, the steepness of the slope actually. And what we show here is that in the countries that are prescribing more ARBs, a higher percentage of individuals that are using ARBs had a lower first 3-week mortality than countries that uses less, that individuals uses less ARBs. And this is really a very crude analysis. I mean, there are a lot of confounding, and therefore we wanted to use another sort of model for trying to show the same thing. And what we did here is that we calculated whether countries would use a higher potent ARB, a higher percentage of potent ARB as compared to a lower potent ARB. And the potency of the ARB was we define that as the affinity to the receptor. So for instance, telmisartan is a very high potency ARB. It has a really strong affinity for the AT1 receptor, and it is a half-life of almost 24 hours, whereas valsartan and losartan have a very low affinity for the AT1 receptor and has a half-life of around 6 to 12 hours. So that is really a different kind of class of ARB. And if you also do the same association with the first 3-week mortality, you can see that countries that are prescribing high potency ARB have a lower 3-week mortality than countries prescribing a low potency ARB. So that suggests that there's also a kind of dose control, control status. The really recent analysis we did was looking at what happened if in hospitalized SARS-CoV-2 patients, if you would discontinue their ARB medication. And we saw that if you do that, if ARB medication was discontinued in the hospital, that there was a 2 times higher risk of dying. And it was corrected for the severity of disease with which the patients were admitted. And this data will be presented by Sandeep Singh at 16:55 in the same meeting. So if you are interested, you can hear his presentation. So to conclude, why is a SARS-CoV pandemic actually a problem? Well, it is a problem because it is a high mortality rate among the elderly. About 8% of individuals over 70 years and about 15% of individuals over 80 years. There is no innate immunity. That means that there will be a lot of sick people, severely ill people, and this will cause a large burden on the healthcare system. And actually the most importantly is that the threat it poses to the routine healthcare system. And for now, we only have effective measures of infection control, but no real treatment. And we believe that ARBs might be, and especially telmisartan, an effective and safe and most of all cheap treatment option to be considered. So I would like to acknowledge some of my co-workers, and I would be happy to take any questions.